Welcome back. In the past, we've already discussed fasting and autophagy. However, science has made significant progress since then. For example, 16-hour fasting is no longer considered the undisputed gold standard, and it's becoming apparent that the timing of food intake might play a more crucial role than the fasting window itself. Stay tuned, because today we're going to dive deep into the fascinating world of autophagy and present you with the latest findings. Autophagy, this ingenious recycling process of our cells, a mechanism that breaks down and reuses damaged parts, essentially the body's own garbage disposal and rejuvenation cure in one. In 2016, Yoshinori Ohsumi even received the Nobel Prize in Medicine for it. It's a fundamental process for our cellular health, and we know that our lifestyle, especially fasting, can influence it. But how did we even discover it? The discovery of autophagy was a kind of cellular detective work. As early as the 1960s, researchers like the Belgian Nobel laureate Christian de Duve observed vesicles in cells under the electron microscope, which apparently enclosed parts of the cell's own interior and transported them for degradation. He coined the term autophagy, literally self-eating. However, the real breakthrough came in the 1990s through Yoshinori Ohsumi and his team. They used baker's yeast, a simple yet powerful model organism, and through clever experiments identified the crucial genes that control this process, the so-called ATG genes, autophagy-related genes. That was the key to deciphering the molecular blueprint of autophagy. Why is this process so ingenious and universal? Evolutionarily speaking, autophagy is an ancient survival mechanism. We find it in almost all eukaryotic cells, from yeast to humans. This shows it is fundamentally important. It likely evolved to help cells survive periods of starvation by recycling unneeded or damaged parts to gain energy and building blocks. But how does it work molecularly? Simply put, when the cell receives stress signals, like nutrient deficiency or damaged components, a machinery is set in motion. A special membrane, the phagophore, begins to form and expands to enclose the cargo, be it an old power plant, mitochondrion, or clumped proteins. A closed vesicle, the autophagosome, is formed. This package is then transported to the cellular recycling yard, the lysosome. After the fusion of the two, aggressive enzymes in the lysosome break down the contents into their basic building blocks. These valuable molecules are then released again and can be used by the cell to build new structures or to generate energy. A perfect cycle. So, autophagy is much more than just garbage disposal during hunger. Today, we understand that it plays a central role in maintaining cellular health. It is the cell's most important quality control. Damaged components, misfolded proteins, or entire organelles that no longer function properly are specifically recognized and disposed of before they can cause damage. A particularly critical example of this is mitochondria, the powerhouses of our cells. Why is their control so important? Mitochondria not only produce the majority of our cellular energy, ATP, but are also the main source of reactive oxygen species, aggressive molecules that can be useful in small amounts, but in excess lead to oxidative stress and damage cellular structures. When mitochondria age or become damaged, they become inefficient. They produce less energy and often even more harmful reactive oxygen species. They can also release signaling substances that initiate programmed cell death. The timely and efficient removal of these defective power plants is therefore absolutely crucial to protect the cell from damage and maintain its function. And for this very task, there is a specialized process, mitophagy. Why isn't it just called autophagy? The key difference from normal or bulk autophagy, which rather non-specifically grabs and breaks down parts of the cytoplasm, lies in its selectivity. Mitophagy is a highly targeted mechanism. Damaged mitochondria are marked by specific molecular signals, like a kind of "please dispose of" label. Special cargo receptors on the autophagy machinery recognize these marked mitochondria and ensure that only they are enclosed by the autophagosome and delivered to lysosomes for degradation. So mitophagy is the targeted autophagy of mitochondria. Similarly, there are other specialized forms of autophagy, for example for peroxisomes, hexophagy, or ribosomes, ribophagy. If this specialized cleaning, like mitophagy or general autophagy, doesn't function properly, dangerous cellular waste accumulates, and this is a hallmark of many age-related diseases, including neurodegenerative diseases like Alzheimer's or Parkinson's, where the accumulation of defective mitochondria and the resulting oxidative stress demonstrably play an important role. Furthermore, autophagy not only provides energy during nutrient scarcity, but can also help defend against pathogens by neutralizing invading bacteria or viruses. It even plays a role in developmental processes and the immune response. Well-functioning autophagy, including its specialized forms like mitophagy, is therefore increasingly seen as a key to healthy aging and the prevention of chronic diseases. Now that we understand what autophagy is and how important it is, let's return to the question, how can we influence it through our lifestyle? And that brings fasting back into focus. Recent years have been marked by a real hype around intermittent fasting, especially the 16-8 method. Many studies, often on rodents, showed great effects, but research on humans as of 2025 paints a more nuanced picture. It turns out, many of the observed benefits of time-restricted eating seem to be simply due to a reduction in calorie intake, not necessarily a magical effect of the time window itself. And the big question has always been, how long do you have to fast to really kickstart autophagy? The answer in 2025 is still not set in stone, but we have clearer indications. Significant autophagy processes in humans seem to begin after about 12 to 16 hours of food abstention. Activity continues to increase and often peaks after 24 to 48 hours. Therefore, it still holds true that longer fasts, that is, over 48 to 72 hours, can potentially increase autophagy rates the most. But, and this is a big but, longer fasting also carries risks. The concern about muscle loss is real, especially with multi-day fasts. Although the exciting Genesis study, with results expected in 2025, shows that with a 12-day fast with very low calorie intake, approximately 250 kilocalories per day, muscle loss can be minimal and reversible, likely through protein recycling via autophagy. But this is an extreme scenario and not suitable for everyone. Therefore, the trend in 2025 is moving away from rigid, daily fasting rules towards more flexible and perhaps more sustainable methods. The new minimum. Many experts see 18 hours of fasting, 18-6, as a more effective minimum, as important physiological processes only really get going after about 16 hours. 16-8 is now considered by some researchers to be more like just skipping breakfast. Burst fasting, a newer approach. Here, you fast intermittently, for example 18 hours, for 3 to 5 days at a time, followed by 3 to 5 days without fasting. The idea, periodically shock the body to maximize fat burning and counteract metabolic adaptation, while also incorporating recovery phases. It's about alternating between the sympathetic, stress, and parasympathetic, recovery states. Fasting Mimicking Diet, FMD, developed by Dr. Valter Longo. A 5-day regimen with a greatly reduced, plant-based, low-protein calorie intake. It's designed to mimic the effects of fasting, including autophagy, without having to completely abstain from food. A gentler but scientifically studied method for cellular cleansing and potentially slowing down aging processes. Perhaps even more exciting is the insight into meal timing. Research in 2025 suggests that it's less about how long the eating window is, but rather when it is and what you eat. The body seems to process food better in the morning and at midday. Our insulin sensitivity is higher then. Late, heavy meals, on the other hand, can massively disrupt sleep quality as the body is busy with digestion at night instead of repair. The recommendation is clear. Focus on breakfast and lunch for the main calorie intake. A light, early dinner. This not only supports sleep but also autophagy, which is more active during the nightly fasting hours. Besides fasting itself and the correct timing of meals, there are, of course, other levers we can use to support autophagy and our cellular health. Exercise remains a key factor. Research increasingly emphasizes the importance of resistance training, especially on days with intermittent fasting. It sends a strong signal for muscle preservation and can even stimulate autophagy in the muscles itself. Endurance training is also valuable, possibly at moderate intensity or on non-fasting days, to avoid overstraining the body. A bonus, training in a fasted state seems to further enhance autophagy signals. Regarding nutrition, there are two important aspects. First, protein is king for muscle protection. When we fast or skip meals, we absolutely must ensure sufficient protein intake during eating phases to preserve valuable muscle mass. Second, certain foods can be supportive. So-called SIRT foods like green vegetables, berries, or turmeric, as well as spermidine-rich foods like mushrooms or nuts, can activate enzymes, sirtuins, or influence autophagy pathways. However, they are helpers, not a substitute for the basics. And how do carbohydrates fit into the picture, especially in the context of our focus on meal timing? The basic rule for effectively initiating nightly autophagy remains. A light, early dinner is ideal for quickly lowering insulin levels. This generally speaks for a low-carbohydrate last meal. Now, there's an interesting discussion about whether a very small amount of carbohydrates in the evening could improve sleep and thus indirectly promote nightly regeneration and autophagy. Here, one must weigh. Do you sacrifice the quickest possible insulin drop for potentially better sleep? This is individual, and the amount would be absolutely critical. We're talking about minimal portions as part of an otherwise early and light dinner. For most, however, it's probably most effective to stick to the light and early dinner rule and place carbohydrates at earlier meals when insulin sensitivity is higher. Stress management and sleep are non-negotiable. Fasting itself is a mild stressor. If a lot of external stress is added, it can become too much. Then it might be better to adjust or pause fasting. Good. Sufficient sleep is fundamental. During this time, the most important repair and cleanup processes, including autophagy, run at full speed. Anything that disturbs sleep, like late heavy meals, hinders this process. Lastly, a few more helpers. Heat exposure through sauna or hot baths seems to promote autophagy via heat shock proteins. Caution is advised with cold exposure during fasting. The combined stress load could be too high for some. Unsweetened herbal teas like green tea or yerba mate are usually unproblematic during the fasting window. But stay away from diet sodas. Artificial sweeteners are suspected of negatively affecting metabolism and even gut flora, which can counteract the fasting goal. And how do you measure all this? An important lab marker is the so-called LC3B lipidation. LC3B is a protein that specifically binds to the membrane of recycling vesicles, the autophagosomes, during autophagy. This specific change, the lipidation of LC3B, is measured, for example, in white blood cells, like neutrophils. The more of this modified LC3B binds to the vesicles transporting cellular debris to the lysosomes, the higher the autophagy rate. We can measure this in the lab. It shows that fasting increases autophagy, but for home use, this is of course not feasible. The direct, simple measurement of autophagy in humans remains complex. However, indirect markers like ketone levels can provide clues. In summary, fasting, when applied correctly, can be a powerful tool to stimulate autophagy and potentially promote cellular health, reduce inflammation, and improve metabolism. But in 2025, we know it's not just about duration, but about the method. Burst fasting, fasting mimicking diet or alternatives, and the timing of meals. Muscle protection through resistance training and sufficient protein is crucial. Stress management and sleep are fundamental. It is important to note that fasting is not a one-size-fits-all solution and may not be suitable for everyone, particularly those who are pregnant, have eating disorders, or suffer from certain medical conditions. Always consult a healthcare professional if you have any uncertainties. Furthermore, a widely discussed study from 2024 that linked intermittent fasting to cardiovascular risks faced significant methodological criticism from experts, but it's highlighting the need for caution against making broad generalizations. So, what's the take-home message? Be flexible. Fasting should support your life, not control it. Focus on when you eat, ideally earlier in the day. This timing might be more important than the mere duration of the fasting window. Find a rhythm that suits you and is sustainable, whether it's 16-8, 18-6, or another variation. Prioritize protein and exercise to protect your muscles, and above all, listen to your body. We hope this update has given you new perspectives on fasting and autophagy. It remains an incredibly exciting field. If you have any questions, feel free to write them in the comments. As always, we wish you a long, healthy, and happy life. Until next time.